spend about 20 minutes discussing some of my lab's recent work in the realm of our area of major interest, which is how do tumor suppressive mechanisms that control genome integrity actually operate in the pathogenesis of cancer, and can they be potentially exploited for early intervention in cancer? At the outset, let me say that all the work that I'm going to share with you was carried out at the Medical Research Council's Cancer Unit in Cambridge. And the mission of the unit, since its founding some 15 years ago, has been perhaps different from many other cancer centers in that we're interested in finding new ways to intervene early in the disease rather than late. My own interest in this problem started with an observation that the clinicians in the audience will instantly recognize. And that is the very unusual fact that if you look in most epithelial tissues, only a very small fraction of pre-malignant lesions ever become malignant. And this ranges from perhaps 5% to 10% in most tissues in which common cancers originate. So the question is, what happens there to prevent the evolution of cancer? And it's pretty obvious that there must be homeostatic mechanisms operating in normal tissues whose job it is to suppress carcinogenesis, and that it is the breakdown of these mechanisms that is necessary for allowing escape and progression of premalignant lesions into frank cancer. So when I started my lab up at the MRC in Cambridge many years ago, we thought that we would look at inherited early onset cancer syndromes. In other words, patients who have a genetic mutation that makes them susceptible to cancers arising at a much earlier age than usual. The logic being that these gene mutations are likely to affect genes which function as critical mediators of these homeostatic mechanisms. I'm going to take just a brief digression to give you some background information. Apologies to those who are already familiar with these ideas. If you take most common epithelial cancers in humans, about 5% of them show a clear familial predisposition, and about 90 to 95% of them show no such predisposition. They're the so-called sporadic cases. At the time I became interested in this problem, there had already been a lot of efforts to try to identify genes that predispose to early onset breast cancer in families, and indeed the genes BRCA1 and BRCA2, which most of you will be familiar with, were identified in the mid to late 1990s through such efforts. It's since become clear that besides these high penetrance cancer-causing mutations in BRCA1 and BRCA2, familial cancer risk in tissues like the breast also occurs through mutations in several other genes, some of which confer moderate to low penetrance risk. But please note that there is this large fraction, nearly 50% of risk, that still, as of 2018, is unattributed. We don't know why these families have an excess of breast cancer predisposition. So there's a lot of work to do. Let me also emphasize that in studying these types of conditions, the insights we gain are not restricted to rare genetic diseases. In fact, it's become abundantly clear in the breast as well as in many other tissues that somatic alterations that occur in sporadic cases of cancer affect the very same processes that are altered in people who carry these high penetrance mutations. So with that said, let me turn to BRCA2. As I said, it was one of the first genes identified, mutations in which confer not only a high predisposition to breast cancer and ovarian cancer, but also to cancers of many other epithelial tissues, the pancreas, the prostate, and so on. So it's not just a breast cancer gene. The protein encoded by BRCA2, as you can see in this cartoon, is ginormous. 3,418 amino acids in the mouse. Moreover, the protein sequence contains no similarities to proteins of known function. So when we started to analyze its function, we really were starting from ground zero, if you will. But long story short, my lab discovered many years ago that inactivation of BRCA2 precipitates spontaneous instability of chromosome structure and number during cell division, And that spotlighted for us the maintenance of genome integrity as a key process necessary for preventing cancer progression and initiation in human tissues. What do we now know about BRCA2 and how its functions prevent cancer? BRCA2 has been attributed many functions in cell biology, but what I'm going to focus on is functions of BRCA2 that relate to the repair of DNA damage or DNA lesions and structures that occur during cell division, during the process of replication. And let me draw your attention to this particular process, homologous DNA recombination, which is a means that mammalian cells use to repair double-stranded DNA breaks, which, as you know, are amongst the most toxic lesions that a cell can encounter during division. Homologous recombination is an error-free mechanism to repair these types of breaks, and it works when a flush-ended double-strand break is cleaved by nucleases to expose long overhanging single strands of DNA, which are then coated with this enzyme depicted here in green, RAD51, which is a recombination enzyme. Now, those of you who have sharp eyes will have seen in the previous slide that I showed within the middle of BRCA2 8 short repeated peptide motifs. These are called the BRC repeats, and their job is to bind to the RAD51 enzyme and not only to deliver it to its sites of activity at double-strand DNA breaks, but also to regulate the activity of RAD51 during DNA repair. And this appears to be a major function for BRCA2 involved in the maintenance of genome integrity. The work of many labs, including my own, has revealed, I think, a pretty good picture of how BRCA2 works in this process. With apologies to the many workers who have contributed in this area, I'm going to focus on findings from my own lab for lack of time. So it's our first set of experiments was focused on trying to reassemble recombination reactions mediated by RAD51 in vitro using single-stranded and double-stranded DNA substrates and recombinant RAD51 and fragments of BRCA2. And what we found is depicted for you on the left-hand side in this cartoon. Essentially, what we found was that in the absence of BRCA2, RAD51 promiscuously engages with double-stranded DNA rather than single-stranded DNA. And you'll immediately appreciate that means that the reaction in which single-stranded DNA coated with RAD51 invades the homologous duplex simply will not occur efficiently. What BRCA2 does through the BRC repeats that bind RAD51 is to reverse the order of these steps. It stabilizes the RAD51 engagement with single-stranded DNA while simultaneously inhibiting the double-strand DNA RAD51 filament. These activities, these opposing activities, if you will, enable the reaction to proceed efficiently. How does it work? At the structural level, we still don't know. But what I'd like to acquaint you with is some of the clues that are revealed by recent structural work in my own laboratory. And let me start by showing you on the right-hand side of this, in the upper panel, a monomeric crystal structure we solved some years ago of a complex between the core catalytic domain of RAD51, shown to you in pink and magenta, and one of the 8 BRC repeats of BRCA2, shown in green. You will see in that picture, which is a sort of ribbon diagram, that there are 2 main sites for the BRC repeats to engage with RAD51. One of them you'll see at that nice little 3,5 beta hairpin that opposes 2 beta strands one in RAD51, one in BRCA2. And in the panel below, you will see an extended alpha helix that also engages the surface of RAD51. So 2 binding sites. We have not, despite years of effort, been able to resolve the structure of RAD51 multimers on DNA using X-ray crystallography, so we have recently turned to cryo-electron microscopy. And what I'm showing you here are initial results at about 4 to 5 angstrom resolution of RAD51, 3 RAD51 protomers encasing a single strand of DNA in the center. But let me draw your attention to the 2 panels shown on the other side. What are the 2 interactions of BRCA2 with the RAD51 multimer? We found that there is both an inhibitory interaction as well as a permissive interaction mediated by these 2 segments of the BRC repeats. You'll see at the top that the inhibitory interaction is actually positioned at the RAD51 protomer-protomer interface. So if BRCA2 interacts at that inhibitory site, RAD51 cannot polymerize. The second interaction, the alpha helical interaction, is by contrast permissive. It's compatible with filament formation because it engages the side, not the protomer-protomer interface in the filament. What we now think is that this gives a clue as to how BRCA2 regulates recombination. As I told you, biochemically, BRCA2 promotes the single-strand multimer, inhibits the double-strand multimer. And we think that this is all down to the difference in the pitch of RAD51 on single-strand and double-strand DNA. In the double-strand filament, there is a compaction of RAD51 protomers, an alteration in the protomer-protomer geometry, and we think that this enables the inhibitory engagement of the BRC repeats. This cannot happen in the extended form of the single-stranded filament, and this differential activity, we think, suggests a structural mechanism for how BRCA2 does this mystical process of differentially promoting or inhibiting RAD51 structures on 2 kinds of DNA. So I want to turn from the analysis of BRCA2's function in homologous recombination to perhaps a more salient question connected with cancer, pathogenesis, and early intervention. How does BRCA2's functions in homologous recombination, which I've shared with you here, actually work in suppressing genome instability and preventing cancer pathogenesis? Like many people in the field, I thought until about 5 years ago that BRCA2's functions in tumor suppression were all down to its role in DNA repair by homologous recombination, until Ferdinando Skoulidis, a clinician scientist in my lab, found using a mouse model that recapitulated the features of familial pancreatic cancer in BRCA2 mutated families, that this was not the case. The crux of the paradox is this. We found not only in the mouse model, but also in tumor samples from human BRCA2 mutation carriers, that BRCA2 violates the Knudsen 2-hit paradigm for tumor suppression. In other words, heterozygous mutations in BRCA2 suffice for carcinogenesis. But we also know that these heterozygous mutations in BRCA2 have no measurable effect on homologous recombination. So how does BRCA2 heterozygosity promote tumors? To share with you some of our recent data in this area, let me start by saying that there are hundreds of known mutations in BRCA2, and only a fraction of them are unambiguously associated with cancer predisposition. About 70% of these are truncating mutants in the gene, and about 30% are missense mutants, which do tend to cluster in particular regions of the gene. We first started by asking about the missense mutations. And as you'll see here, I wish I could show you, but I'll just have to point, that there is a cluster of mutations in BRCA2 that change residues at a key stretch of BRCA2 that engages with a cognate partner protein called DSS1. And what we were able to show is that these mutations prevent the interaction of BRCA2 with DSS1, and the failure of this engagement results in the exposure of a cryptic nuclear export signal. This results in the mislocalization of Rad51 BRCA2 complexes out with the nucleus of the cell, and crucially, these missense mutations act as dominant negatives. They mislocalize BRCA2 Rad51 even in the heterozygous state when wild-type BRCA2 is present. And we think that's how this important cluster of missense mutations works. What about truncating mutants? I'm showing you here two common truncating mutants in BRCA2 that are pathogenic. How do they work? I'm going to cut straight to the chase, knowing that I'll probably run out of time, and tell you the answer. We have, to our great surprise, found recently that mutation carriers for these truncating mutations suffer from, we think, a gene-environment metabolism interaction, which is what predisposes them to cancer. And the culprit here are aldehydes, which you know are pervasive in our environment and are also generated by all sorts of metabolic processes endogenously in cells. Why did we come to this conclusion? Well, we were first able to show, and this is the work of Sean Tan and Sakshi Chadha, that BRCA2 heterozygous cells exposed to aldehydes but not to other replication blocking agents suffer from severe replication tract instability. And this is depicted for you here in these analyses using DNA fiber combing of the average length of replication tracts. For lack of time, I can't take you through all the details of the experiment, but suffice it for me to say that there is a selective effect of the aldehydes in inducing replication tract shortening, not in wild-type cells, but in BRCA2 heterozygous cells, and this effect is not seen with many other DNA-damaging agents. As many of you in the audience are aware, about 500 million people worldwide carry a mutation that inactivates the acetaldehyde catabolizing enzyme ALDH2. They have excess production of acetaldehyde after exposure to ethanol, and we're able to show that acetaldehyde in our cellular systems has the same effect as other aldehydes. How does this work? The key finding is we are able to show that aldehydes have a previously unrecognized effect in selectively depleting about 30 to 50 proteins detectable by swath mass spectrometry in the entire cellular proteome. It's a very selective effect. And you can see that BRCA2 is one of the proteins targeted by this effect. Aldehydes deplete BRCA2 from cells. And what happens when BRCA2 deficient cells or BRCA2 heterozygous cells are exposed to aldehydes is that they are highly sensitive to this degradative effect, and that induces a haploinsufficient phenotype. This, we think, is a kind of interesting way by which tumor suppressor mutations may interact with the environment and with metabolic processes to confer disease risk. The idea is that BRCA2 heterozygous mutation carriers are perfectly normal, but they have lower amounts of BRCA2. When exposed to aldehydes, induced proteolysis pushes the BRCA2 protein levels below the functional threshold, and this makes these carriers exquisitely sensitive to the effects of aldehydes. This also got us, for the first time, some notoriety in the mass newspaper, The Sun, which some of you may recognize. As you can see, The Sun translated our findings into the following catchy phrase: booze, shampoo, car fumes, all cause cancer. That's not the case. We are still studying the mechanism, but we have some interesting observations now which give us a likely culprit. Many of you will know that unscheduled hybrids between RNA and DNA occur normally during cell division and many cellular processes like transcription. Andre Aguilares' lab showed a couple of years ago that these hybrids accumulate in excessive amounts in BRCA2 deficient cells, but didn't show how or what this would do. In a nutshell, we were able to show in the same work that an enzyme that digests R loops in BRCA2 deficient cells also ameliorates typical genomic instability, suggesting that this is the cause or a cause of genome instability following BRCA2 inactivation. So where do these R loops come from? Very recent work in press from Mahmoud Shivji, Xavier, and Cheatham in my lab reveals an unexpected source of these R loops. We find that BRCA2 has a new function in gene transcription. As you know, RNA polymerase II in most metazoan genes initiates, but then after about 50 to 60 nucleotides pauses for a while before entering rapid elongation. We find that BRCA2 regulates this switch, and that in the absence of BRCA2, that slow switching results in the accumulation of R loops at the promoter proximal pausing sites of many genes in the genome, and this leads to double-strand DNA breakage via the enzyme ERCC4, ERCC5. So we now have another function of BRCA to think about, its function in transcription as a potential source of DNA damage and genomic instability in BRCA2 mutation carriers. My time is up, so I'm going to end with one last point, which is, I hope that I have illustrated how our work on the mechanism underlying inherited cancer predisposition by BRCA2 mutation has given us some insights into two questions highly relevant to early intervention in cancer. Who amongst us is most likely to develop cancer, and can we stratify those at highest risk in order to enact preventive measures? I'll end with some work unrelated to BRCA2, which I think also illustrates the point that some of this information on tumor suppression may help us to prevent or even treat cancers at an early stage. And this is the idea that genomes get scrambled as tumors evolve. And this genomic scrambling rewires biochemical pathways in such a way as to make therapy of late-stage disease extremely difficult. Is there a way to intervene early? We've been working for some time on cells which have no other abnormality other than the activation of the oncogene mutant KRAS. And very recently, as I've shown there, we have found in vitro and in mouse models in proof of concept experiments that it is possible to intervene by accentuating an Achilles heel induced by mutant KRAS. Mutant KRAS induces mitotic abnormalities, and we have been able to target these mitotic abnormalities to sensitize RAS mutant cells at a very early stage in their development using inhibitors of protein-protein interactions of mitotic kinases. So I'll end there and say I'm very hopeful that this type of work promises new approaches for early intervention in the clinic in the not too distant future. Thanks for your attention.